Hi, everybody. My name is Caitlin, and we are here today at Behind the Scenes of Science. The goal of this webinar series is to give you a sense of some of the careers in science, different pathways, how do you go from high school to a career in science, and what does an average day look like in the career of people who work in and around science. So today, we have Melissa Hendershott, a scientist in the Allen Institute for Cell Science. If you have any questions for her at any time during the webinar today, please use the Q&A in Zoom or the YouTube chat to go ahead and ask those. And I'm going to start with some questions to get us going, and then we will have plenty of time for questions from you. So to get us started, can you tell us about your science journey from high school to now?
Yes. So like Caitlin said, my name is Melissa Hendershott. I'm a scientist at the Allen Institute for Cell Science. And I think I've been doing science for probably 15 years now. I remember back when I was in high school, I think I had a family friend who gave me some book that was like careers to do if you're interested in science. And it was things like doctor or nurse or engineer. And I was like, but I just want to do science. Like science is so cool. And I just want to be doing research. And that's not listed as a career in this book. So it took me a little while of kind of learning and talking to people before I sort of learned that you could just do scientific research as a career, and that the path to doing that was to go to graduate school and learn how to do, or one path to doing that is to go to graduate school and do research there. And so basically, as soon as I figured out that that was an option, I was like, oh, that's the thing that I want to do. So when I was an undergraduate, I went to Boston University and I started volunteering in a lab and, said, I just want to learn how to do some molecular biology techniques, and started learning that way. And that was a really great gateway into being able to do undergraduate research. And then, I loved doing that, and that kind of set me up to be able to apply to graduate school, and ultimately get my PhD. And I think that set me up for where I am now at the Allen Institute.
What did you study as an undergrad and in grad school?
Great question. So my undergraduate degree is in biochemistry and molecular biology. So I took a bunch of chemistry, I took a bunch of biology classes when I was in college as an undergraduate. And then in graduate school, I did, I think my degree is in cell biology, which,
Yeah, in grad school, the title on your degree is much less important than the work that you do in lab. Can you tell us more about what you worked on as a grad student?
Yeah. So I think one of the things that I have really loved about doing cell biology in general, and I think my PhD work and the work I do at the Allen Institute in particular, is that I've done a lot of imaging and a lot of microscopy, which means that basically every piece of data that I have ever collected or looked at is basically a picture of something that's happening. So when I was a graduate student, I was studying these structures in cells called microtubules. So microtubules are sort of these structural elements of cells. They're almost like steel beams in a building, so they help give structure to the cell, and they're also pathways for things to get transported around and moved around in cells. But they're really cool structures because they're dynamic, and so cells use them. They assemble them and they disassemble them. So when a cell is dividing, they make this structure called the spindle. That's what actually pulls DNA apart. But when a cell isn't dividing, when it's just an interphase, the microtubules are just sort of extended all over the cell. And so this structure is really dynamic so that cells can quickly rearrange it and remodel it and use it for different purposes. And so the question that I was sort of studying when I was a PhD student was, how do cells regulate the dynamics of these microtubule structures? And the way that I was doing that was using this really cool kind of purified system. So it turns out that you can take tubulin, which is the little pieces that make up microtubules. You can purify those from cells, and then you can actually put them in a test tube and they will make a microtubule in the test tube. And so you can actually image that process while it happens, and then you can add in other proteins. So I was studying these particular proteins that bind to one specific end of the microtubule, and you would actually watch those proteins bind to the end of the microtubule, and you could see how that changed how these structures were assembling in this, artificial test tube system that we could study under the microscope. So anyway, that was basically what I studied in graduate school, and it was really, really, really cool to see these structures that, you read about in cells and you knew were really complicated, and to see that behavior recapitulated on a microscope in this single molecule way was really cool. It was the best thing about the research that I was doing at the time.
That's fantastic. So you mentioned that the majority of the data that you've ever collected has been in the form of images. So when you're collecting that data and analyzing it, what does an average day look like for you, both now and pre-pandemic?
It's a great question. So I spend basically, I would say, equal time both being in the lab and, literally just sitting in front of a microscope and getting it in focus and setting up an acquisition and collecting data. And then I spend basically the same amount of time once I have a bunch of data going back through and analyzing it and trying to figure out what do those pictures actually tell me about what this process is doing. So some days that looks like, I'll be on the microscope for an hour and then I'll go back to my desk and kind of try to review the data and figure out what was happening and plan a new experiment. Sometimes it's, an experiment will be more complicated and it will take me a whole day to collect the data. And then I might need a couple days in a row of sort of sitting at my computer and making sure that I've analyzed and understood what's happening in the experiment. And to be honest, that changed a little bit when COVID happened. We were home for a few weeks and it was honestly really nice to have some time to just sit and really look at this, enormous amount of data that we had collected before COVID and have time to really sift through it and figure out what was going on. Now we're really lucky to get to be back in the lab a few days a week. And so, we basically just plan really carefully so that we have not very many people in the lab at a time, but we can go and get our experiments done and collect the data and then, again, still have that time to go back and actually understand what we captured.
So the microscopes that you're describing are not the microscopes that I think most high schoolers would have seen. Can you tell us about what those microscopes collect? How do they work? How is their function and operation different from, say, a standard light microscope?
Yeah, so what's cool about microscopes is that even though the microscopes that we use in the lab at the Allen are more complicated than the light microscope that you might have encountered at school, at the end of the day, they're all still using sort of the same optical principles, which is pretty cool. If you've ever used a microscope at school and you can look through the oculars and you turn the light on and you put your sample on and you get it in focus, we go through that exact same process when we go to set up an acquisition on one of the microscopes that we use at the Allen. But we have a couple of sort of special things about the microscopes that we use that let us collect the kinds of data that you can see in my background here. So one thing that we get to have are lasers on the microscope. And so that allows us to do what's called fluorescent microscopy. So the cells that we get to image at the Allen Institute for Cell Science they've undergone this process called gene editing. And so basically what that lets us do is it lets us put a molecule that glows when you shine a laser on it. We attach that to a particular protein. And so what that lets us do is it lets us light up that protein. And if that protein localizes to a particular structure in the cell, which is most of the things that we label, you can shine a laser on those cells and then you can just see that structure in the cell. And what's extra cool is you can actually do dynamic imaging and you can image over time and see how that structure maybe changes as cells divide or as cells move around. And so you can get this dynamic data about the cell. So that's one thing that's cool that we get to use. We have lasers. And then the other thing that's really sophisticated and kind of advanced about the microscopes that we have is that we have those microscopes attached to a computer. And we have these programs that let us really quickly make adjustments to things about the light path of the microscope. We can change the lasers that we're using so we can look at different structures in the same cells. And we're able to say, I would like to image these 10 positions, and I would also like to image them every 5 minutes for the next hour. And so being able to sort of have that automated control through the computer lets us collect really complicated data sets that let us look at a lot of different kinds of behavior in the cells that we're studying.
And these are human cells in a dish. Can you tell us a little bit more about those cells?
Yeah. So we work with this particular kind of cell called an induced pluripotent stem cell. So basically what that means is that at some point in the past, somebody actually donated some of their skin cells And we took those cells, and some scientists who are not at the Allen Institute, but some other scientists, basically took those cells, and instead of them being skin cells, they reprogrammed them to be stem cells. And what's cool and kind of special about a stem cell is that it can become lots of different cell types. So we can actually maintain and kind of grow those stem cells in a dish, but we can also, if we give them the right signals, we can make them become lots of different kinds of cells. And so one of the projects that I work on a lot at the Allen Institute is a project where we take those stem cells and we actually give them signals that let them turn into cardiomyocytes. And so that's the kind of cell that you're seeing in my background here, is a stem cell that's been told to become a cardiomyocyte. And a cardiomyocyte is a cell, it's sort of an immature version of the cells that are in your heart. But one of the things that's super cool about working with them is that they actually beat the same way that the cells in your heart would beat to pump blood around your body. And so we can study that process. How does a stem cell actually change into a cardiomyocyte? What changes happen in the cell? What's going on in that process? But it's cool because we can study it in dish and we can actually image lots of parts of that process.
Great. What's your favorite thing about your job?
That's a great question. And I would say I have two favorite things. One thing that's awesome, I think about working at the Allen in particular, but I think about the direction that science is going in general now. Science is much more of a kind of team endeavor. So there are lots of us working on the same problem. And so, one of my favorite things about my job is that I work with really incredible people, and it's really awesome to be able to solve scientific problems together as a team. The other thing that I love about my job, I love that the data that I collect and that I get to look at is image-based data. I think there's something really incredible about wanting to understand a process and see what's happening, and to be able to have this visual data where you can look at a picture that describes this process that you want to understand is really, really incredible.
That's awesome. It's great that you get to work with the kind of data that is your favorite every day.
Yes.
And for those of you who want to check out that data, it's all available at allencell.org. So you have your favorite things about your job, but sometimes things don't always go exactly right or as planned. What has been your best science failure?
Yeah. So my best / biggest science failure happened to me in graduate school. So in science, there's this sort of goal that you want to be studying something that's new. So a lot of scientific research is really oriented towards asking a question that no one has ever asked and kind of being the first person to figure out the answer to that question. And everybody wants to be the first, everybody wants to be doing something novel and exciting, but sometimes, multiple people will be working on the same question. And sometimes you know about it and sometimes you don't. But if one person gets there first and there are other people who are working on that same question and they kind of get there second or later, there's this word for that, which is called being scooped. And so when I was in graduate school, the project that I was working on got scooped by another lab. And I got scooped in this sort of particular way that we had really independently done almost the exact same experiment. And so, this other lab published this paper. It was the first that I knew that they were working on this. I had been doing my work. It had all been going fine. And the thing that happened was we actually found the exact same thing, which is sort of remarkable that these two labs working completely independently got to the same answer to this scientific question that we were both interested in. And so I got really lucky, actually, that my boss at the time, Ron Bale, who was my graduate advisor, helped me get my paper published, even though this other lab had gotten this result first. And I think the thing that I actually think was really cool about that, so in addition to just meaning that I was able to get my PhD, I was able to finish graduate school. I think it's really important in science. It's good to know that two people got the same answer, because it means that you're much more likely to have been right if you both got to the same answer. It's sort of like if you take a test and your friend got the same answer to this calculus question as you're pretty sure that you probably both did it right. It would be weird if you both did it wrong in the same way. So I actually think, in some ways it's sort of too bad that more of science doesn't work that way, that that we can't kind of work, more in teams and kind of check each other's answers. But I think it's pretty cool that that was actually how it worked out in my project.
That is pretty cool. Well, I see we have a few questions that have come in from the audience. As a reminder, if you do have questions, please use the Zoom Q&A button to ask those questions. Or if you're watching on YouTube, you can just put them into the YouTube chat. So the first question is, what are your feelings on the implications of gene editing and how it can be used for right or wrong?
That's a really, really good question.
And a big question.
It is. It's a big question. I think from my perspective, I think CRISPR is really one of the most incredible scientific tools that has come along. Really, I think GFP, the green fluorescent protein, so the protein that we use to tag the structures in our cells, I think that was a huge, huge development in the whole field of bioscience. And I think CRISPR is an even...
What does GFP enable that it makes it so special?
That's a great question. So GFP allows us to do, this kind of microscopy, this fluorescent microscopy that I was talking about earlier. It allows us to do that kind of microscopy, and it allows us to put a specific tag on a particular protein or structure in a cell and just look at that one specific thing. And that was something to be able to do that on a microscope and be able to image cells in real time. That's something that has really completely changed the kinds of questions and the kind of research that we can do in bioscience. And I think CRISPR is a really similar kind of great leap forward for the field of scientific research. And that's not even talking about any of the therapeutic implications. And I think that's where you start to get into this question of how can we use it for good and how can we use it for bad. So I absolutely think it's a tool that's going to change. I mean, it has already changed the way that we do scientific research. And I think it will continue to do that. And I think there's lots of unanswered questions about how it might be used in therapeutics in the future. There's a documentary that a friend of mine helped work on called Human Nature. And I think that's one of the best documentaries I've ever seen about what the implications of CRISPR could be for human health and kind of the future of bioscience. So I think if you're interested in learning more, that's my favorite thing that I've ever seen. I definitely recommend checking that out.
Thank you for the recommendation. We have another audience question. And as a reminder to the audience, please go ahead and put those into the Zoom Q&A or YouTube chat. Are induced pluripotent stem cells the most prevalently used stem cells in research today? And also, I'll add to that, what makes the induced pluripotent stem cells special? Why are they so useful for the work that you're doing?
Yeah, those are great questions. I would say that, yes, pluripotent stem cells are the most commonly used stem cell kind of in scientific research. They are great because they're relatively easy to work with. There are things about them that are harder to work with than some other kinds of tissue culture systems. But in general, the induced pluripotent stem cells are pretty easy to maintain in a lab. You can passage them for a long time, which basically just means we can keep a population of cells going in a dish and pull cells out to do experiments with, but we can have this regenerating population of cells that we can work with in the lab. In addition, one of the things that's really, really cool about induced pluripotent cells is that you can generate them from pretty much any person who's alive walking around today. You can make stem cells from their skin. And so one of the things that lets us do is if you have a patient who has a particular disease, you can make stem cells from that patient's skin, and it'll have the same genetic makeup as the person who has that disease. And so what it allows you to do is have a whole battery of, maybe 10 patients who all have the same disease. And you can see if you're trying to develop a drug, you can apply that drug to all 10 of those stem cell lines, and you can see if they all respond in the same way or if there are different things. There are things about their genetic makeup that maybe cause them to respond differently to a therapeutic. So because of that, it's a really, really awesome tool for being able to explore these maybe different genetic effects that you might see in different patients with the same disease.
So the Allen Institute for Cell Science works in basic science research. What does that mean for you specifically in the context of working in cell biology and imaging?
It's a great question. So we, because we do basic research at the Allen Institute, I think for me, the thing that means is we're trying to sort of help build the foundation of scientific research. So instead of being focused on developing a particular therapy or studying a particular disease, we're really trying to say, at the foundational level, what new things can we understand about how cells behave in their healthy state? And so I think that's one important thing. Right now, the main cell line that we do most of our research on is a healthy stem cell line. And so we really want to understand what normal cell behavior looks like. So for instance, when we take the cells and make them into cardiomyocytes, what does that process look like in a normal cell? And maybe someday we'll expand into what does that look like in a cell that maybe has some sort of genetic mutation or disease. But we're really trying to understand, just at the basic level, how does the cell behave? What does a healthy cell do when we put it in different environments or subject it to different kinds of perturbations?
What do you wish more people understood about your work? I know CRISPR is a hot topic right now, so.
Yeah, that's a great question. And I think one of the things that I think is really important for people to know and understand about basic science research in particular, so sort of building off of the previous question, is that, just because, sorry, lost my train of thought. Just because we're not focused on a particular disease or creating a cure for cancer or some other disease that people may face, that foundational basic scientific research is something that really allows, the whole scientific community to take these results and take these things that we're learning and apply those to research that is going to help change people's lives. I think one of the things that's like, CRISPR is a great example of this. Actually, CRISPR came out of super basic science research. And I think, it was this mysterious thing that people were seeing in bacteria, and people were really confused about what was going on. And nobody was looking for a way to do gene editing and be able to, revolutionize biology. It was really basic science research that somebody saw a weird thing and they were like, I want to know what's happening. I want to understand what is causing this to be a thing. And that was what ended up leading to CRISPR. And I think that's one of the things that I wish people understood more about basic science research, that it's just really hard to predict what is the weird, cool thing that you're going to find that's going to lead to the next big thing in advancing our understanding of science and biology. And I think that's one of the things that's amazing about biology is nature has figured out so many ways to make life be robust and be able to continue. And so you just never know what you're going to find. And that's one of the things that I think is really exciting about being in biology and being a scientist. And I think it's also the reason that basic science research is really, really important, because you just never know where you're going to look and find something really cool.
That's wonderful. I totally agree. We have another question from an audience. So this is a basic science question. Do viruses have cells? So for example, for COVID-19. And I'll expand on that question a little bit. How does a virus interact with a cell?
That's a great question. I'll start with a disclaimer that I am not a virologist. There are lots of people whose full-time job is to study how viruses work. But in general, scientists, biologists do not think of viruses as being cells. So most scientists like to talk about cells as having a membrane that goes around the outside of them and sort of makes a bubble that keeps all of the cell parts inside of it. Most viruses do not have a membrane that goes around them. They're really pretty much made of nucleic acid, which is things like DNA and RNA, so the genetic material, and then they're made of protein. So they have these protein shells that sort of encase their genetic material. And the way the virus interacts with a cell is that the virus will land on the outside of the cell and it will figure out a way to get itself inside of the cell. And then the goal of it getting inside of the cell is to basically put its genetic material into the cell, and it will take over the cell's replication machinery and basically use the cell as a tool to help it make more virus. And so that's the that's the number one goal of the virus, is make more virus, infect more cells. And so that's often the thing that makes you sick. You know, the virus will, you know, if you have a cold, it'll infect your throat and it will kill a bunch of cells in your throat, and that's what makes you have a sore throat. But that's part of why that we don't think of viruses as sort of being a cell or being alive in the normal way that we talk about things being alive, because it takes invading another cell for a virus to be able to make more copies of itself.
All right, I have our very last question of the day. So thank you everyone again for joining us today. And we will be back in 2 weeks on November 10th with Anna Marie Yanni, a research associate in our brain science division. And our last question for Melissa today is, when you were a high schooler, what do you wish you had known about a career in or around science?
That's a great question. I think the thing that I wish that I had known was how broad a career in science can be. And I think there are just such an enormous set of ways that you can participate in science, ranging from being a research associate to having a PhD, to being on the business side of science, to being somebody who writes about science, to being an educator. You know, I think, yeah, just the amazing kind of broad set of ways that you can be a scientist or be part of the scientific community is really cool and really interesting. And I think sometimes as scientists, we don't do a good enough job of kind of talking about how many options there are. And so I wish that I had known that as a high school student, and I hope that more people can get exposed to all of the various ways that you can be a scientist.
To follow up on that, you mentioned that you had gotten this book as a high schooler. What do you wish you knew as a high schooler on how to go from being a high schooler to having your job specifically?
That's a great question.
The job title is scientist.
Right. I think one of the things that I sort of wish that I had not done, you know, I think I decided early on in high school that I really wanted to be a scientist, and that was the path that I wanted to be on. And I think one of the things that I would tell my high school self is, you don't have to know, and that it's good to explore really broad interests. And I think if I had said, like, this is my only path, I want to be a scientist at the Allen Institute, I think I probably wouldn't have ended up here. And so I think my biggest advice is, you know, it's good to just be open to opportunities. I think if you're somebody who's curious and somebody who has a background in science, you will have lots of options. And it's good to just be curious and be open to opportunities that might come your way, even if they're not exactly what you thought you wanted to be doing.
Well, thank you so much. And thank you everyone for joining us here today. Again, we're going to be off next week. We will see you on November 10th, where our guest will be Anna Marie Yanni, a research associate in our neuroscience brain science division. And I hope we will see you there. You can watch the recording of this talk today on the Behind the Scenes of Science page soon. We will post it soon. And thank you all for joining us.